So briefly in this talk, I'm going to present like two of my ongoing projects on the Connectome 2. So the first project is about using joint diffusion relaxometry models to improve axon diameter mapping. And the second project is about probing the water diffusion inside the myelin shield. So first about axon diameter mapping. So why are we interested in mapping the axon diameter in vivo? Well, a lot of studies have been showing that the axon diameter is actually related to the conduction velocity in the brain. So if we can measure the axon diameter non-invasively, this will provide us windows to connect the structure to the functional outcome. And previously in our lab, we have been showing that the axon diameter we can measure by MRI is associated with, for example, aging. So by measuring the axon diameter, we may be able to monitor the neurodegenerations. And also in, for example, in multiple sclerosis case, studies have been showing that, for example, here is this histological data. The MS patients have appearing larger axon diameter compared to the control, could potentially due to inflammations, and also they demonstrate some axonal loss. And our group has also been showing that with the MRI data, we also seen kind of this trend with the MRI data. So this will help us to provide a measure to, for example, monitor disease progressions, and also maybe related to the disability of the patient. So we can measure axon diameter with diffusion MRI. There are many models available. So one of the methods that we have been using is called Excalibur SMT. Basically, we try to relate the diffusion signal inside the axons is associated with the axon, the transverse velocity inside the axons is related to the axonal diameter. So the larger the axon diameters, there are different time dependency in the diffusion signal. And using Connectome 1, we have been previously showing that we can capture some of the axon diameter variations in vivo in the brain, for example. And the cortical spinal tract, which usually have a larger axon diameter compared to the surrounding white matter. And recently, we demonstrated that using the high gradient performance with the Connectome 2, we may be able to probe a smaller axon in the brain. And we validated that with the in vivo data, showing that the axon diameter map we obtained from the Connectome 2 is smaller than what we have previously obtained. So while this is very promising, but as Marco mentions in the morning, that it's actually very difficult to measure the small axons in the brain. That is because the diffusion contrast we obtain or we try to measure is proportional to the fourth power of the axon size. And the effective radius is even to the sixth order of the means of the radius. So what does that mean? It means that the sensitivity will collapse for small axons. And for the axons that we try to also measure with diffusions will be weighted heavily on the tail of the large axons. However, if we look in the histology data, it has been showing that most of the human axons are smaller than 1 micron. This is well below the sensitivity limits, even with the state-of-the-art Connectome 2 scanner. Interestingly, there are also another way to try to measure axon diameter. So recent study from the group in Lausanne demonstrated that if we can use diffusions to filter out, for example, the extracellular signal, and we measure the T2 of the intraaxonal signal. So what it shows is that the R2 we measure is inversely proportional to the axon radius. That's very interesting because this provides another contrast mechanism that have an opposite effect on the axon size, because now the smaller the axon size, the stronger the contrast we can get. And by applying this method, they demonstrated that they can also obtain the in vivo axon diameter map, demonstrating similar contrast as we can obtain with the diffusion. So my project is about trying to combine both two contrasts into a single optimizations problem, because the diffusions and T2 are sensitive to the axon size differently. So with diffusions, we can try to measure the diffusivity to the axons, and they're very sensitive to the large axons. Thus, the T2 models might not be reliable. And in the meantime, with the T2 relaxations, because the signal is proportional to the inverse of R due to the surface relaxation effect, so this contrast is actually very sensitive to small axons. So by combining both together, this might give us a comprehensive sensitivity across the full physiological range, not just for healthy tissue, but also for diseased tissue. So our methods, we call this multi-echo Excalibur SMT or Excalibur SMT. So that's involve updating the biophysical model, which have the joint, diffusion, and T2 contrast mechanisms, where the interactional signal encode both the time-dependent diffusion contrast and the surface relaxation contrast. And we acquire multi-echo diffusion MRI data on the Connectome 2 for the demonstrations by simultaneously varying the B value, the diffusion time, as well as the echo time, so that we can measure both the diffusion and the T2 effect across different B values. So here show our preliminary in vivo result. On the left, we have the conventional Excalibur SMT using just the diffusion-only data. And in the middle, we have the Excalibur SMT result. We can see in this image that's the cortical spinal tract, which usually have a large axon diameter. The proposed methods capture these features very well. But if we focus on the frontal white matter, which histological data shown that usually contains more axons, it really demonstrates that the diffusion-only Excalibur SMT lack the sensitivity to measure the small axons there. So there, with the diffusion-only data, the axon radius we can obtain is around like 2 microns. But with the Excalibur SMT methods, the axon diameter we measure there is much lower and closer to the T2 models. So here show another example in the corpus callosum. So here we can see that all methods can capture this low-high contrast across the corpus callosum. But with the Excalibur SMT, this contrast become the strongest compared to the other model. So in addition to provide better sensitivity to small axons, the signal model also mitigate the complemental T2 bias. So here I'm showing the simulations result on the estimations of, on the left should be the intraaxonal fractions estimate, and on the right is the CSF fractions estimate. So without consider the T2 effect, we can see both parameters have overestimations, but when we consider the T2 effects, this can be corrected. And this is supported by our in vivo data, where the proposed methods show a little bit lower near fractions and the CSF fractions estimate. In addition to that, we also obtain an extra parameter on the extracellular T2, which we still need more investigations on what information to study what information it contains. So while the Connectome 2 data show very promising result, what we also interested is about if we can translate these methods to more to other scanner that's more accessible. For example, the Sigma X, which is commercially available, but without the gradient performance is not as strong as the Connectome 2. So therefore, we try to acquire the multi-echo diffusion data on the Sigma X on the same patient, on the same subjects. But to be able to do that, we have to update our acquisition parameters. That include having a longer minimum diffusion time compared to Connectome 2, longer gradient pulse width, lower B values, and longer minimum echo times. So in principle, this suggests us that we would not have this good sensitivity as the Connectome 2. As we can show, here is the comparisons between the Excalibur SMT result on 2 scanner. With the Connectome 2 data, we can clearly see the cortical spinal tract contrast, but such contrast is not so obvious on the sigma X result. And this is also supported by this scatter plot analysis between the Connectome 2 and the sigma X, where it's really the correlations between the 2 scans, even on the same subjects, is very low because of the resolution limits on the sigma X. And with the max caliber SMT result, we can see that both scanners can capture the cortical spinal tract pretty well. And the correlations between the 2 scans are much improved in this case, because now we incorporate this T2 contrast in the diffusion model that really provide the improved sensitivity to measure the small axons. So to switch gear a little bit, I'm going to talk a bit about the myelin water diffusions. So myelin is very important in our brain because it's able rapid neuronal signal and also axonal support. There are lots of methods in MRI that can measure the amount of myelin inside the brain, such as myelin water imaging. But most methods doesn't really tell us about whether the shield is intact or they become lost or not. So it would be nice to have a way to have a more direct access on the myelin shield's integrity. So one way we think might be interesting is to use diffusions because we can measure the diffusions. If we can measure the diffusion inside the myelin shields, though, this might provide more direct markers of the shield's integrity. So recently, Hongxi and I have proposed this theoretical framework on measuring the outer axon diameter using, if we can measure the diffusions inside the myelin shields, which might also provide an estimate of a marker of the outer fiber diameter. So this is interesting because if, for example, if we can combine this with the more conventional axon diameter mapping, this might allow us to have a more direct assessments on the G ratio instead of aggregated G ratio. But here is the problem. So with diffusion, like with standard diffusion imaging at 3 Tesla, we need a long echo time to be able to achieve a sufficient diffusion encoding. So most applications require the echo time of 70 milliseconds, but the myelin water T2 is at 3 Tesla, usually around 20 to 25 milliseconds. So the signal usually is gone before the encoding started. So how can we achieve this in vivo? So here we propose methods called stair DWI, which we incorporate additional adiabatic inversion pulse with short TR with the conventional diffusion module. So in this pulse sequence diagram, we can see that on the right-hand side here, it's just a standard diffusion module, but before we apply a adiabatic full passage pulse to suppress. So the effect is that we can suppress the long T1, T2 intra-extraxonal water while leaving most of the myelin water unchanged. So if we do that, we can focus more on the diffusions on myelin water. And together, if we have the short TE enabled by Connectome 2, we can achieve the shortest echo time of 21 milliseconds with the maximum B values of 1600. And we implemented this sequence to acquire whole brain coverage acquisitions with 5 B shells and 80 directions. So to validate if our signal is really sensitive to myelin water, we acquire several B0 image. So here show just one of them. So here's the B0 image with the echo time of 21 milliseconds. We can see that in this image, the gray matter is highly suppressed in contrast to the white matter. And by acquiring multiple echo times, we can perform a T2 analysis. So what it shows is that the T2 we measure with the sequence is around 18 milliseconds in white matter. So this value is quite comparable to the literature values of the myelin water, which is around 20 milliseconds. So this supporting that what we are trying to measure, at least most of the signal is coming from the myelin water. So then we conduct several, like some analysis to compare, for example, the stair DWI and the standard DWI. We try to keep the acquisition parameter as close as possible. And here show the spherical mean signal between the stair DWI and the standard DWI. And we can see that these two methods have a really distinct spherical mean signal. And so then the first analysis we conduct is to fit the data. We fit the DTI models to the data. So the mean diffusivity suggested that the stair DWI have a much lower diffusivity compared to the standard DWI. We also try to estimate some apparent diffusivity coefficient parameter using our axisymmetric Gaussian model, so that we can focus on the longitudinal and the transverse apparent diffusivity. So what is shown is that we have this the longitudinal apparent diffusivity is much higher than the transversal one. So here show further more bundle-like analysis. And when we compare these results to previous study using a double inversion recovery experiments, we can see that the result we obtained in vivo has is much more anisotropic compared to the DIR experiments. But with the apparent diffusivity values that is perpendicular to the axons is very close to what previously suggested. So at last, we try to correlate our diffusions result with the myelin water fractions estimation. And we see very low correlations between this metrics. So this might suggest that our diffusions metrics might provide complementary information to myelin water fractions, which reflect the myelin content in the brain. So with that, I want to thank the team and my collaborators of sharing the idea generously with me. I'm happy to take any questions. Thank you.